Probability, according to mathematics, is a number, okay? And this is number describe how likely it is that an event will occur, okay? Look out. Monohybrid crosses by using Punnett square is the studying of inheritance of one characteristics such as the color of peas pod, green or yellow, okay? So think back to Mendel's experiment. He carefully counted the offspring from every cross, and when he crossed the two plants that were hybrid from stem height, carry genotype T capital T small, about 3/4 of the second generation had tall stem and about 1/4 had short stem. Each time Mendel repeated the cross, he observed similar results. Mendel was the first scientist recognized the principles of the probability can predict the result of genetic crosses. Okay, now scientists use a Punnett square, where a Punnett square is a chart that shows all possible ways alleles can combine in genetic cross. Or another definition is a diagram that is used to show all possible ways alleles can combine in genetic cross to measure the probability of genetic outcome, okay? This is used by biologists to determine the probability of the genotype that carried by offspring, okay? In genetic cross, the combination of alleles that parents can pass to an offspring is based on probability, okay, not sure. Example, the cross between two hybrid pea plants with a round seed that carry genotype R capital R small, where R capital refer to the round seed, which is a dominant allele, and R small refer to the wrinkled seeds, which is a recessive allele, okay? Let's start by the steps. Step number 1, start by drawing a box and dividing it into 4 squares, as we see, okay? Then the male parents' alleles are written along the top of the square. We write the male parents' allele along the top of the square, and the female parents' allele right at the left side, okay? So each square refers to the offspring resulted from the crosses, from after cross, okay? So each square receive one allele from male parent and other from female parents. So the result will be, as we see in the inner box, okay? And the inner boxes represent the offspring, okay? So by studying the outcome, we note that we have 3 over 4 have round seed, and 1 over 4 only has wrinkled seed, okay? Mendel didn't know about alleles, but a Punnett square shows why he got the result he saw in the second generations, okay? The plants with the alleles R capital, and alleles R capital R small would have the same appearance of the trait, okay? All of them have round seeds. Only plants that carry alleles R small would have wrinkled seeds, okay? So we use another terms, phenotype and genotype. What is the difference between them? Phenotype is a set of observable characteristics or traits of an organism, like the appearance of the traits on organism, like tallness, dwarfness, okay? Genotype is a genetic makeup or alleles, okay? So if we have example, the allele for a small pea pods S capital is dominant over the allele of the pinched pea pods S small. All the plants with the least one S allele, S capital allele, have the same phenotype. the same appearance of trait, okay? All of them produce smooth buds. These plants can have two different genotypes, S capital, S capital, or S capital, S small. And we can't differentiate between them because both have the same appearance, okay? The plants with pinched buds have the same phenotype, pinched buds, as well as the same genotypes, S small, okay? So biologists use two terms to describe an organism's genotype, okay? So we have another term, homozygous and heterozygous. What's the difference between them? Homozygous, an organism that carry two identical alleles for a trait, okay? Like the small bud plant that has the alleles S capital, and the pinched bud S small. But heterozygous, an organism that has two different alleles for the trait, like a smooth bud plant with the alleles S capital, S small, okay? Mendel used the term purebred to describe the homozygous, and used term hybrid to describe the heterozygous, okay? Thank you. Grade 7, and goodbye.